Have you ever wondered how viruses are removed from biopharmaceuticals? This fascinating process is known as viral filtration, and it's designed to remove a wide spectrum of viruses from process streams in biopharmaceutical manufacturing. These viruses could potentially pose a risk to product safety and patient health, making their removal of paramount importance. The types of viruses targeted for removal can vary. This depends on the specific manufacturing process and the regulatory requirements governing the production of the biopharmaceutical product, such as monoclonal antibodies. Now, you might be curious about the types of viruses that are commonly targeted. These can be classified into two major categories: enveloped viruses and non-enveloped viruses. Enveloped viruses, as the name suggests, are encapsulated by a lipid membrane derived from the host cell. Examples of these include viruses like the human immunodeficiency virus, hepatitis B and C viruses, herpes simplex virus, and the influenza virus. On the other hand, non-enveloped viruses lack a lipid membrane and are more resistant to inactivation. These include viruses like adenovirus, norovirus, rotavirus, poliovirus, and parvovirus. So how does viral filtration work? The technology typically involves the use of specialized filters with pore sizes small enough to physically retain viruses, while allowing smaller molecules like proteins and buffer components to pass through. The primary types of filters used for viral filtration in biopharmaceutical manufacturing include membrane filters, depth filters, and nanofilters. Membrane filters consist of a porous membrane material with defined pore sizes that can range from nanometers to micrometers. This operates on the principle of size exclusion. Larger particles are retained on the surface while smaller particles pass through. Depth filters are composed of a matrix of fibrous or granular materials, with a complex network of interconnected pores. They operate on the mechanism of depth penetration, capturing particles within the filter matrix as the process fluid passes through. Depth filters offer high capacity and can effectively capture a wide range of contaminants, including viruses. Nanofilters are a bit more advanced. They're engineered with nanoscale features, such as nanofibers or nanocomposite materials, to achieve high selectivity and efficiency in virus removal. Nanofiltration operates on the principles of size exclusion and electrostatic interactions, removing viruses with high specificity while minimizing the impact on product yield and quality. The selection of the appropriate viral filtration technology depends on several factors, such as the characteristics of the target viruses, the desired level of viral clearance, process requirements, and regulatory guidelines. Manufacturers often conduct validation studies to demonstrate the effectiveness of viral filtration in removing targeted viruses and ensuring the safety and purity of biopharmaceutical products. In summary, viral filtration is a critical process in biopharmaceutical manufacturing, designed to remove a broad spectrum of viruses that could potentially pose a risk to product safety and patient health. It employs specialized filters to physically retain viruses while allowing smaller molecules to pass through. The selection of the appropriate filtration technology depends on a variety of factors, ensuring the safety and purity of biopharmaceutical products.